All right, let's go through it. So, which of the following is the first adhesion molecule upregulated under the action of histamine? So you might remember me talking about Weibel Palade bodies, and those contain pre-synthesized P-selectin, right? P for pre-synthesized. So the first adhesion molecule is going to be P-selectin. E-selectin is made from scratch when you stimulate an endothelial cell with histamine. These other ones don't really get expressed at all. Which of the following is the second phagocytic cell type to migrate from the circulatory system into the area of tissue damage under acute inflammation? So the first group of cells is going to be the neutrophils. The second phagocyte to be recruited is going to be the monocytes. Okay. Which of the following are primary resident cells in tissues that produce IL-1 and TNF during acute inflammation? So that would be the macrophages. Okay. Out of all of these, they're the only tissue cell. Which of the following cells in the extravascular space can produce histamine as a result of trauma to the tissue or complement activation? So histamine, extravascular space, must be a mast cell, right? Basophils can also make histamine, but they're in the blood. They're not in the tissue. Now, which of the following cytokines produced by macrophages are associated with cachexia? That's going to be tumor necrosis factor alpha. The only other option here that are even made by macrophages is IL-8. And IL-8 is just a chemokine for neutrophils and T cells and basophils. TNF is actually like an inflammatory cytokine. Okay, which of the following are considered chemokines for neutrophils and naive T cells? That's going to be IL-8, made by macrophages. Which of the following may be activated by plasmin? This is going to be C3. Remember, C3 has a bunch of ways that it can be cleaved into C3b and C3a, right? Any of the C3 convertases, plasmin, which is like a regulation protein, can cleave C3. Also, IgA can cleave C3, and mast cell and neutrophil proteases can cleave C3. So there's a lot of ways to activate it. Which of the following are primary producers of lipoxins? So this is going to be your platelets. They make lipoxins. That's one of the eicosanoids. Protein kinase C interacting with DAG can lead to the activation of which of the following enzymes present in neutrophils, PMNs? Okay, so in my opinion, there's actually not really a correct answer here, but you can narrow this one down. This is not an enzyme. Neither of these are enzymes. The only two enzymes here are NADPH oxidase and phospholipase C. So you might remember that phospholipase C is the enzyme that cleaves PIP2 into DAG and IP3. So it wouldn't make sense if DAG activated phospholipase C, because phospholipase C is the only way that you can even get DAG in the first place. So the only answer that really makes sense here is NADPH oxidase. It's the only enzyme that could possibly come after DAG. In my opinion, there wasn't a correct answer in this one, but that would be the best answer if I had to pick one. So the answer to number 10, where it says the immediate outcome associated with activation of the correct enzyme from question 9 would be which of the following? NADPH oxidase produces reactive oxygen species. It produces superoxide anion from oxygen. Vasculitis associated with immune complexes usually occurs most frequently in which of the following tissues? So this is going to go with the type 3 hypersensitivity stuff. And that's going to include glomeruli of the kidney and the sinusoids of the liver. Okay, the brain is not usually going to be a place where immune complexes cause damage. Sinusoids of the heart, there are no sinusoids in the heart. And the left ventricle of the heart is definitely quite large. It's not small enough for immune complexes to deposit there. So that's not going to be one of your answers. But the kidney, the glomeruli are fenestrated, and so that makes kidneys a usual target of immune complexes. And then sinusoids in the liver have, they're like little holes in the vascular endothelium where immune complexes can end up getting lodged. So that's type 3 hypersensitivity kind of stuff. Which of the following terms below would best be associated with moving between endothelial junctions? That's going to be diapedesis. That's when cells move between endothelial junctions. Okay, all these other ones have nothing to do with that. Okay, and then there's a bunch of complement questions. So if you didn't study complement, you should have studied complement. So which of the following components are involved in the classical pathway? This one's multiple answer. So classical pathway starts with C1q. C1q cleaves C1r. C1r cleaves C1s. And C1s cleaves C4 into C4b, which binds to the pathogen. C2 then binds to C4b, and it's cleaved by C1s into C2b. And then that serves as a C3 convertase. So C3 is going to be involved in the classical pathway. The fact that it's multiple answer tells you that at least 2 of the options are correct. Okay? So if it's multiple answer, at least 2 of them are correct. None of these other ones are involved. These are all involved in the alternative pathway. Okay, which of the following complement components are zymogens? So a zymogen is an enzyme that needs to be cleaved in order to become active. So we're looking for an enzyme here. C1 is not an enzyme, or I guess it technically is. C2b, so C1s and C1r are both zymogens, right? So I'm thinking this is probably the answer. C2b technically is an enzyme, right? But C2b has already been cleaved, so it's no longer a zymogen, it's already the active enzyme. So that's not going to be your answer. So C2b has already been cleaved from C2. It's no longer a zymogen. C3 is not an enzyme, and C10 is not even a protein in complement. IgA bound to a target can activate which complement component? That's going to be C3. IgA can cause C3 to be cleaved into C3a and C3b. Okay. Which complement component comprises part of the C3 convertase for the classical pathway? So the C3 convertase in the classical pathway is composed of C4b, C2b. So your answer here is going to be C2b. Which of the following are termed anaphytoxins? So any of the A components can potentially be anaphytoxins. A for anaphytoxin, right? So C3A there. And then this was a highly missed question. Which of the following stabilizes the alternative pathway C3 convertase? That's going to be factor P, or pridin. This is the only positive regulator of complement. The only positive regulator of complement is factor P. None of these other ones have anything to do with that. Which of the following on binding a target initiates the classical pathway? That's going to be C1, right? C1q binds to the target, and then that triggers the classical pathway to happen. Which of the following on binding a target initiates the alternative pathway? C3b is the answer to that one. You might have chosen C3, but C3 is not actually going to bind to a target, right? C3 has to be cleaved before it will bind. So C3 itself does not stick to anything, it's just a serum protein. Once it's cleaved into C3b, however, then it will bind to something, right? So C3b is what really binds to the target to initiate alternative pathway. Now, which of the following can cleave C4? That's going to be C1s. Remember, in the C1 protein, it's QRS. They're in alphabetical order, in the order that they get turned on. So C1q binds, that turns on C1r. C1r cleaves C1s, And then C1s can cleave C4 and C2. All right. Which of the following is a function of inositol triphosphate, IP3? Remember, IP3 is inositol triphosphate. Activate G proteins? Nope. Production of heats? No, that would only be the case if we were talking about like arachidonic acid or something like that. Influx of calcium from outside the cell? No. Release of calcium from the ER and mitochondrion? That's going to be the best answer. Induce respiratory burst? No, that's not going to be one of them. So remember, there's an IP3 receptor on the endoplasmic reticulum, and it serves as a calcium channel. I actually don't know that the mitochondrion is a place where calcium gets stored. So I don't know if that particular part of that answer choice is right, but the fact that it says from the ER makes this the best answer choice, in my opinion. I actually am not familiar with whether the mitochondrion is a place for IP3 to bind and release calcium. But I definitely know that the ER is a place where calcium is stored. Okay, which can activate C1s? So remember, it goes QRS. So C1R is going to activate C1s. QRS in that order. Okay. C-reactive protein can promote binding of which? That's going to be C1q. C1q, its ligands include IgG, IgM, and C-reactive protein, right? And also potentially some other stuff, but those are the three important ones. Which can function as opsonins or opsins? So opsonins, these are things that stick to surfaces to mark them for phagocytosis. Remember that the B components are what serves as opsonins, especially C3b and C4b. Those are your two biggest opsonins in complement, C3b and C4b. Which polymerizes to form pores in lipid bilayers? That's going to be C9. C9 polymerizes 10 to 16 molecules of it will insert into the membrane in a pore to open a hole. Which of the following lymphocytes have CD4 as a surface molecule? So this is going to be any kind of T helper cell. T helper cell. So Th1 cell is a subset of a T helper cell. So Th1 is the only one here that is a T helper cell. Which of the following lymphocytes use surface-bound immunoglobulins as an antigen receptor? That one is going to be a B cell, right? An IgM molecule or an IgA molecule or an IgD molecule bound to the surface of the cell is the B cell receptor. And we did talk about that. Which of the following can present antigen in the context of MHC2? This is going to be any of your antigen-presenting cells. There's 4 of them. Macrophages, monocytes, dendritic cells, and B cells. Okay? So the only one listed here is a B cell. The only one there is a B cell. Which of the following are phagocytes found primarily in extravascular tissues? This one did not have a correct answer listed whatsoever. Sanders told me he was going for macrophages, but he forgot to include it as an option. So there was no answer to this one. You got it right automatically. Which of the following cells have CD8 on their surface? This is going to be a cytotoxic T cell. They respond to MHC1. Which of the following lymphocytes produce IL-10? IL-10 is only made by Th2 cells. It's also made by Treg cells. Okay, but Th2 cells are the classic IL-10 producers. And IL-10 basically makes macrophages stop making so many inflammatory cytokines. It tells them to turn into M2 macrophages, and IL-10 also suppresses the Th1 response. Okay, which of the following cells can develop into plasma cells? That's going to be your B cell. Okay, B cells turn into plasma cells. Plasma cells are the terminally differentiated B cells that just make a bunch of antibody for 3 to 5 days before dying. The plasma cells have a short lifespan, usually, and they are the terminally differentiated form of B cells. They don't revert back to any of their previous developmental stages. Okay, which of the following lymphocytes, so there's a key word right there, lymphocytes, can bind to stress indicators on cells, and unless a secondary signal is transmitted by their KIR, they will kill the target cell. We definitely talked about this a few times. The KIR is the killer cell inhibitory receptor, right? This is going to be your natural killer cell. So natural killer cells, they look for stress indicators on cells, and they're going to get all angry and they're going to get murderous unless the cell is expressing MHC1. And the KIR is actually a receptor for MHC1 in most cases. So MHC1 inhibits natural killer cells, and that occurs through the KIR receptor. KIR is a don't kill signal located on natural killer cells. Which of the following lymphocytes produce chemokines that recruit polymorphneutrophils, or PMNs, or neutrophils? So this was another highly missed question. This is going to be Th17 cells. None of the other ones produce chemokines that recruit neutrophils. Probably some people chose Th1 cells. That would be my guess. My only other guess as to which of these might seem right. Th1 cells, they make a cytokine called IL-3, and IL-3 does technically, it spreads to the bone marrow, it diffuses the bone marrow through the blood, and when it gets to the bone marrow, it promotes the production of white blood cells in general, but Th1 cells don't make chemokines of any type. They do not produce chemokines. Okay, Th17 cells are the only lymphocyte here that makes chemokines. Dendritic cells are not lymphocytes. Dendritic cells are myeloid cells, right? They're phagocytes, not lymphocytes. Lymphocytes are T cells, B cells, and natural killer cells and ILCs, the innate lymphoid cells, right? So dendritic cells are not included in that. Which of the following lymphocytes recognize antigen by an epitope-specific receptor and can produce interferon gamma? So we're looking for a lymphocyte with a specific receptor, so a T cell or a B cell, that can produce interferon gamma. That's going to be Th1 cells. Okay. Which of the following cells recognize antigen presented in the context of MHC1? That will be cytotoxic T cells. They express CD8 and they respond to MHC1. T helper cells, they express CD4 and they respond to MHC2. Okay. Which of the following immunoglobulin classes is the most common immunoglobulin class on mucosal surfaces? That's going to be IgA. Okay. IgA for that one. IgA is the most concentrated on the mucosa. Which of the following cytokines promote T cell and natural killer cell proliferation? This is going to be IL-2. IL-2 is a growth factor for all lymphocytes, especially T cells and natural killer cells, but also it can act on B cells as well. Which of the following cytokines has a comparatively long half-life and can trigger acute phase proteins from the liver? This is going to be IL-6. IL-6 has a long half-life compared to the other cytokines that are made. IL-1 technically can trigger acute phase proteins from the liver, but you need a whole lot of it because it dissipates really quickly. It's broken down in the blood really quickly. So it doesn't have a very long half-life. You're going to need a lot of it in order for it to reach the liver to any significant extent. Okay, so only IL-6 for this one because it has the long half-life. Okay. So, any questions on those? I know this went rough. Hopefully you get an opportunity to learn how to study better and know what know what Dr. Sanders' mind works like a little bit better, the first exam is usually pretty rough. But I'm going to tell you right now that the second exam is not exactly easier. So make sure you use this as feedback for your studying strategy for the next exam. If you did bad, there's probably something you need to change, um, whether that be procrastination or studying the wrong material, or maybe you didn't go to SI as much as you wanted to. Use feedback for yourself. Those are just some common things that people usually say. Okay. We're going to start the exam 2 stuff now. The first content for exam 2 is applied immunology. Okay. So applied immunology seems hard, but once you kind of get the hang of it, it's actually not that bad. So this is just a list of everything in here. The appendix might be a good place to consult. So it's called the Immunologist's Toolbox. If I was going to tell you to read one chapter, I would probably say to read Appendix 1. So do that. Focus on the stuff that we cover in this class, though, because there's a lot of extra stuff in there. This PowerPoint is an exhaustive list of everything you need to know. So how to approach applied immunology. Reading Appendix 1, the relevant parts of it. The trade-off for applied immunology is that you can't really find PubMed articles like you could for a lot of the other stuff, but you can find some of it on YouTube. A lot of people are going to be on YouTube explaining how to do these kinds of assays and what happens and stuff. So sometimes videos can be helpful. I know Sanders relies on videos for some of the tests. He has them in the PowerPoint. So what to make note of when taking notes and studying applied immunology is, what can the test be used to do? What is the end game or purpose? Usually, you're looking for an antibody using antigen, or you're looking for an antigen using antibody. In other cases, you're trying to create a therapeutic, or you're trying to create an antibody of some kind. And occasionally you might be, separating cells based on what proteins they're expressing or something like that. A lot of these are just fishing for antibody or antigen using a known antibody or antigen. Okay. How do you run the test step by step from beginning to finish? That's going to be a good thing to know. If you can answer question 2 for each test, you're pretty much golden. What is the logic behind each step? That can be a good thing to know, but because you don't have any essay questions this semester, number 3 is probably not as high yield. What would you get as a result of each step? Like what's in your flask or what's in your well or whatever you're working with? And when would you run this test instead of another test? Maybe it's more appropriate for certain functions. But maybe it lets you analyze more proteins at once in an easier way. Maybe more pros, less cons. Some of them are more expensive than others and take longer to do. Sometimes that's a pro or con. Number 5 is probably less high yield than the other ones, but if you can do number 2, you're like golden. You're going to need to know a couple of different reagents as well. So this would be for like an example of a studying table or a spreadsheet if you're keeping one of those. I personally didn't make a spreadsheet for this material, but this was also my hardest content at the time. So I wish I would have made a table or spreadsheet for myself. So most of these assays are looking for an antigen using an antibody, or they're looking for an antibody in serum using a known antigen. Okay. So how do antigens and antibodies actually get held together? There's 4 to 5 ways this can happen. So this will be a review of cell biology and probably general chemistry as well. So first of all, electrostatic forces hold together charged groups that are opposite from one another. on neighboring molecules, right? That's one of, it's all going to be non-covalent, right? Antibodies bond non-covalently to antigens. So electrostatic forces, also hydrogen bonds, you know, a hydrogen attached to a high electronegative atom comes into contact with a high electronegative atom on another molecule. Van der Waals forces, typically that's associated with hydrophobic molecules. You get little fluctuations in electron clouds in brief windows of time. Hydrophobic forces, of course, staying away from water because of the van der Waals forces holding them together as well. So hydrophobic forces, van der Waals forces are one example of that. And then a new one that they added in this edition of Janeway is the cation pi interaction. So whenever you have an aromatic ring, like a benzene ring, you know, you get electrons moving very rapidly around in a circle through that ring, and it creates like a swell of negative charge surrounding the ring, so going up and down from the planes of the ring, whereas in the plane of the ring there's like a positive charge. And positively charged or partially positively charged atoms on neighboring molecules can bind to that by electrostatic interaction. So those are the 5 ways that antigens and antibodies can be held together. They're all non-covalent. So that they're non-covalent, especially. And there's some basic terminology that I wish someone had told me before I tried to learn all of this. I'm going to admit to you that Sanders doesn't do a perfect job explaining applied immunology content. I thought it was really confusing when I took the class. So I'm going to do my best to de-scatter it for you. So some early terminology that you should know right away. A supernatant is a fluid remnant within a tube after centrifugation, right? So you centrifuge a tube with cells inside or something like that. The cells all go to the bottom in a pellet or a precipitate, and they're at the bottom because they're heavier and the centrifuge spun them outward. The supernatant is the fluid remnant on top of the pellet, okay? Now, the word wash is going to be used to indicate rinsing stuff out of a mixture with an inert solution like tris PBS or phosphate buffered saline. Tris phosphate buffered saline, right? So, for example, we might want to wash out excess antibodies from a mixture once our antibodies have bound to their antigen. Elute. Elute is an important word. It means to detach specifically bound molecules from one another. For example, you might want to detach an antibody from an antigen so that you can use it to do something else after getting it to bind to antigen. For antibodies, usually to elute them from antigen, you're going to use a glycine buffer at pH 2.5. And you can also heat the mixture to get the antibodies to come off of the antigen. Now the glycine buffer, the major reason that it is used for elution is that the pH is so low. It's so low that it changes all the charges on the antigen and antibody to be positive, and then they repel each other, and the antibody comes off the antigen when that happens. There's also a term that you should know. So goat anti-mouse IgG. This is just an example of some of the nomenclature for antibodies. Goat anti-mouse IgG would mean that you have antibody from a goat, usually IgG from a goat, that binds to the Fc region of mouse IgG molecules. So anytime I say anti-something, I mean that my antibody is binding to that something. So when I say anti-mouse IgG, I mean it's an antibody that's binding to mouse IgG molecules. Okay? Typically, in applied immunology, we're working with IgG molecules. They're the easiest to produce, and they're also sort of the easiest to work with and the most common things available on the market. I mean, it's super cheap to buy a secondary antibody that binds to IgG. So IgG is usually the class that we're working with in applied immunology. Sometimes IgM is what we're looking for, but we're typically going to use IgG anytime we're fishing for an antigen. Affinity is the strength of a single interaction between one binding site and one ligand. Okay? Avidity is the total strength of all combined interaction between a protein's binding sites and all of their ligands. For example, mannose binding lectin in the lectin pathway of complement, it has low affinity for a single mannose moiety, but if there's repetitive mannoses in a complex, the avidity is very high. So it tends to not bind to one mannose, but many mannoses all in a row. That's an example of avidity. Also, 2 binding sites per IgG molecule means that IgG has higher avidity than affinity, right? Because the affinity is only one of the binding sites, but the avidity is all of the binding sites altogether. So IgM, it has 10 binding sites because it's a pentamer, right? And there's 2 molecules per monomer, or there's 2 binding sites per monomer. So it has 10 total binding sites, meaning IgM has a lot of avidity for ligand, for antigen. Has a lot of avidity, but IgM is actually usually lower in affinity than IgG because it doesn't undergo somatic hypermutation. An adjuvant, this is relevant for vaccines. An adjuvant is a substance that delays the release of antigen from a vaccine dose. Usually, this has the effect of strengthening the immune response to the antigen and allowing for better uptake into antigen-presenting cells. It may also, the adjuvant in some cases, may also contain immunostimulatory components. For example, Freund's adjuvant, which is typically used in mouse immunizations, contains proteins derived from Mycobacterium tuberculosis. I think it actually just contains like shredded mycobacteria. And all of those things, the MTB pathogen contains, it contains PAMPs that stimulate PRRs and therefore allow for an inflammatory response by the innate immune system. So you're going to get better co-stimulation expressed on dendritic cells. You're going to get a better T cell response. You're also going to get a better antibody response because T cells are going to be a little bit angrier, and so are T cells when you add an immunostimulatory component. So we'll look at a couple of adjuvants. A positive control is something that you're going to run when you do a test. It's a control condition that simulates a positive test result or test reaction. Okay, so for example, if we're looking for an antigen, we're going to include a positive control of that antigen in a separate reaction. A negative control is a control condition that simulates a negative test result or reaction. You want to run these both to make sure that your assay is working and that it's specific for what you're looking for, right? The negative control can be used in some cases to quantitatively screen out background noise. So a positive result minus the negative controls result tells you what your real signal is. So here's a couple of adjuvants. You don't need to know all the adjuvants, but the common theme among them is that they delay the release of antigen. Okay. Now, adjuvants, they vary from adjuvant to adjuvant what they do and what they are. In humans, usually we use alum, which is an aluminum hydroxide gel. And that just sort of helps invoke macrophage uptake. It delays the release of antigen, obviously, because it's a gel, which doesn't dissolve as easily as just injecting you with a liquid made in water. But in humans, we use alum as the adjuvant, typically. There's some other adjuvants that are used in humans, but a lot of the most common ones are just aluminum hydroxide gels. Now in mice, they often use Freund's adjuvants. Now, incomplete Freund's adjuvant is an oil in water emulsion. Basically, you throw some oil in with your antigen mixture, and then you swish back and forth 1,000 times, and eventually they just emulsify each other. complete Freund's adjuvant contains dead mycobacteria that stimulate C-type lectin receptors, which leads to the induction of co-stimulators in macrophages. So that's one of the more common. These are two of the more common mouse adjuvants. These ones toward the bottom are newer developments for adjuvants. For the most part, in humans, they don't really add anything that's going to get a really strong inflammatory response because we want to avoid causing harm to people, especially with vaccines being such a controversial thing. Maybe we shouldn't appeal to the crazies, but we try not to put anything too upsetting in a vaccine because we don't want everyone to be crazy sore, and that can reduce vaccine how many people take our vaccine, right? So usually alum is good enough. Occasionally maybe you might add something else, but usually try to stay away from anything that might cause an extreme inflammatory response. And it's kind of unfortunate though, because that can decrease the efficacy of a vaccine when you don't use a strong adjuvant. But it is what it is. Two other terms to know: active and passive immunization. Active immunization is immunization that is generated by the recipient or by the patient or whatever. Active immunization is typically very long-lived, so it's going to last years typically, maybe even the whole lifetime of the individual. Active immunization can be achieved through vaccination or through natural infection. So you can be immune to influenza either because you were infected with influenza or because you were vaccinated against influenza. Then there's passive immunization, which was basically generated by something or somewhere else, and that is short-lived. So this often comes in the form of injecting antibodies into people. For example, convalescent antiserum that has been used within the last couple of years to treat COVID. They take antibodies from someone who had an infection with COVID, and they inject those purified antibodies into a patient who currently has COVID, and those antibodies bind to the virus and prevent it from infecting more cells, right? Gamma globulin is another one. Gamma globulin is just IgG molecules that have been purified from a mixture. And IVIG is another one that can give passive immunization. IVIG stands for intravenous immunoglobulins. IVIG is actually a medication used in both outpatient and inpatient settings. IVIG is a big pooled collection of antibodies from the general human population. So it contains antibodies against pretty much every common pathogen that the human race is exposed to. It's a big pooled collection of antibodies. That's what IVIG is. And it is infusion. You cannot give antibodies orally. They have to be injected because they're destroyed the stomach, and they also don't absorb intact across the gastric epithelium. Monoclonal antibodies are another example of passive immunization. This is a chemically pure antibody that has been produced in mass by cells in culture, usually for the purpose of treating or diagnosing a disease. They are directed against a specific antigen, and they are monoclonal, which means they have only one amino acid sequence. Okay, they're not polyclonal. Antisera, like horse antisera. So horse antisera or mouse antisera or whatever. You might want to create a rattlesnake antitoxin by injecting rattlesnake venom into a horse or into a mouse or into an animal of some kind, a goat. And then that animal is going to produce the components of that toxin you injected the animal with. The only issue with this is animal antibodies don't look exactly like human antibodies, so they can cause an allergic response. immune cells can take them up, and then there are T cell epitopes in the antibody that T cell receptors can bind to once they're loaded into MHC2. And then it's a really good candidate for a Th2 response because you don't have typically a lot of it floating around. So you can get an allergic response to that. And the downside of passive immunization is that it doesn't always work because sometimes it can cause excessive inflammation and it can do more harm than good in some cases. But in other cases, it definitely has been shown to be a safe and effective thing for treatment of certain diseases. We have to test it first, but a lot of times it is pretty safe. So here's the deal with serum versus plasma versus whatever. If you collect blood in a vial and then you let it just clot for a while, it's going to break into two pieces. It's going to break into a darkly colored clot that's pretty much solid to gelatinous, and that's going to sit at the bottom of your tube when you centrifuge. And then it's going to break into serum, which is a fluid component. Usually serum is yellow. In this case, there's some red blood cells left here. The serum contains the antibodies. The clot just contains platelets and clotting factors and red blood cells. In other cases, you add an anticoagulant called EDTA, which is a calcium sequestering molecule. You add that to the collection vial, and then if you spin down the red blood cells, the fluid portion is going to give you plasma. Okay. Now, in that case, the plasma still contains clotting factors because no clotting has happened. The clotting factors are still there. So coagulation depends on calcium. So when you add the EDTA, it prevents the clot from occurring. And then when you spin it down in the centrifuge, the red blood cells end up at the bottom, the plasma ends up at the top, and then you get a buffy coat between the two that is composed of white blood cells and platelets. Serum, on the other hand, is when you let it clot and then you spin it. Okay, so serum equals plasma minus the clotting factors. So that's a little bit of background on serum versus plasma. Both of them come from the blood. Okay. And then here is a therapeutic approach that was sort of an early thing that they tried. This is one of the earliest things that was used for like cancer immunotherapy and stuff way back when. Lymphokine activated cell therapy, or LAC, LAC. In most cases, they used IL-2, and it was used for cancer treatment. The process worked like this. They would extract lymphocytes from blood or from the microenvironment surrounding a tumor. They would inject IL-2 into that extraction. So they would stimulate the lymphocytes with IL-2. That would cause those lymphocytes to grow. And then they would transfuse the lymphocytes back into the patient. Now, after this was shown to be effective and was shown to have anti-cancer activity, they started just directly injecting IL-2 into the area of the tumor to help the T cell response fight the tumor. And also interferon gamma was injected into the area surrounding tumors. Both of those are lymphokine activated cell therapies. Okay, so there's some figures here, but this right here is really all you need to know. And then there's this thing called serotyping, which is an antibody type of approach. Serotyping has been done for a good long time. Serotyping is when you ask, what cell surface antigens are there on a cell? For example, on an E. coli cell, typically they look at the O antigen and they look at the H antigen, which is on the flagellum. So O157 is the 157th discovered O antigen from E. coli, and H7 is the 7th flagellar antigen that they discovered of E. coli. And O157H7 is the strain of E. coli that is known to cause enterohemorrhagic E. coli. So if you isolate E. coli from a patient and then you add antibodies to O157 and H7 and the antibodies stick, you can confirm the diagnosis of EHEC, enterohemorrhagic E. coli, in that patient. You've serotyped what strain they're infected with. Now, these codes don't necessarily indicate the proteins that the pathogen uses to cause disease. They are just codes for cell surface antigen. So they don't necessarily tell you like what the bacterium is making to make the person sick, but they're just cell surface antigens. And sometimes they correlate with certain diseases. In this case, O157 and H7 correlate with enterohemorrhagic E. coli. Another example is the Lansfield groups, which is used for Streptococcus versus Enterococcus, and it's also used to characterize Streptococci into different species. So the Lansfield groups are involved in, they allow you to categorize streptococcal bacterial strains and species based on the carbohydrate antigens on the cell surface. So if the Lansfield A antibody binds to the cell, then it must be a Lansfield group A bacterium. But serotyping is not only done on foreign cells. There's also blood typing, which you've probably heard of. Blood typing is a form of serotyping that is based on antigens on red blood cells. They look at the A and B antigens, which are co-expressed if the person has both. You can have type O blood, which you don't have A or B on your red blood cells. You can have only A, or you can have only B, or you can have A and B on your red blood cells. So you can have type O, type A, type B, or type AB. Okay. They also look at the rhesus antigen. This makes you Rhd positive or Rhd negative. So when you hear A negative blood, it means that they have only the A antigen and they don't have the rhesus antigen or the B antigen on their red blood cell. On the other hand, if you hear AB positive, that means they have the A antigen, they have the B antigen, and they have the Rhd antigen, which is probably the third most relevant of all of these, right? There's some other rhesus antigens. There's A, B, C, D, E, all the way down to like G or something. So antibody to A and B, they are made before the person is ever exposed to foreign A or B. Okay, so let's say you have type B blood. If you have type B blood, that means you have anti-A antibodies in your serum because those are made naturally. Okay, so antibody to A and B are going to be present regardless of prior exposure. However, you're not going to make antibody to B if you have type B blood or if you have type AB blood. Okay, you're only going to make you're only going to make antibodies against antigens that are not on your red blood cells. And for A and B, those can be present regardless of prior exposure. On the other hand, antibody to the Rhd antigen only comes after exposure. And this can result in disease. Sometimes a mother will have a child with a father who has the Rhd antigen on his red blood cells, but the mother does not have the Rhd antigen on her red blood cells. What that can result in is a baby who has the Rhd antigen on its red blood cells, and then during birth, the mother is exposed to a little bit of the baby's blood, and that can result in the mother creating an immune response against the Rhd antigen. The next time around the mother gets pregnant, if the child has the Rhd antigen, the fetus is going to be terminated by the mother's antibodies. This is why they give RhoGAM prior to giving birth, so that if the mother is exposed to any of the baby's red blood cells, she will not develop antibodies against the Rhd antigen. That allows us to avoid what we call the hemolytic disease of the newborn, HDN, which is a sort of autoimmune type thing. And it results in the baby basically dying. On the other hand, you don't have to expose somebody to an A antigen in order to get the A antibody. Okay. So let's look at some blood typing. So type O negative blood is the universal donor because the red blood cells that are being donated do not have any antigens on their surface that are immunogenic. Okay, there's no antibodies that can be made against type O negative blood. On the other hand, type AB blood, type AB blood, the donors can only give blood to type AB recipients. That's because if you gave type AB blood to somebody with type A, then the B antigens are going to be attacked by anti-B antibodies in the recipient. Okay? So you can't give any antigens to somebody who doesn't already have that antigen on their red blood cells, if that makes sense. Okay, so on the practice exam, there's some examples of this. The other thing you want to look at is the RHD antigen. You don't want to give RHD positive blood to somebody who doesn't have the RHD antigen on their red blood cells. Okay. Now, on the other hand, when you are an AB positive donor, you may only be able to give blood to AB positive people, but you can give serum to anyone. And that is because your serum is the universal serum. You don't have any anti-A antibodies, any anti-B antibodies, or any anti-RHD antibodies in your blood. Right? You're not going to make antibodies against anything that is on your red blood cells. You only make antibodies against things that are foreign, that are on other people's red blood cells. Okay? So AB positive donors can only donate to AB positive people their red blood cells, but they can donate serum to anyone because they don't have any antibodies that will attack the red blood cells in the recipient. All right? Hopefully that makes sense. Now on the flip side of that, type O negative blood, the red blood cells can be given to anyone, but the serum can only be given to type O recipients because it's going to contain anti-A and anti-B antibodies, and it may also contain anti-RHD antibodies depending on prior exposure. Okay, so hopefully that makes sense. The universal red blood cell donors can only give serum to one type of person, whereas the universal red blood cell or the universal serum donors can only give red blood cells to one type of person. Look at this chart and try to make it make sense. Hopefully it does. This is Janeway's version of it. The O antigen is actually technically an antigen. It's just not immunogenic. So you're never going to make antibodies against this. And everybody has at bare minimum this chain on their red blood cells. But in people who have A, they also express this moiety. And then in people who have B, they also express this moiety on the end. People with type A, B, they have both. Okay. And then the O antigen is going to be present regardless of your blood type. Okay. All right, so that was blood typing. And then there's this thing called fluorescence activated cell sorting, or FACS. Fluorescence activated cell sorting is also called flow cytometry, except it has a little bit more of a function than flow cytometry. Here's how it works. Basically, monoclonal antibodies specific to cell surface antigens, like CD4, are added to a cell suspension. The monoclonal antibody that you use contains a fluorescent dye that shines at a certain UV wavelength. So that monoclonal antibody fluoresces under UV light due to a fluorescent dye attached to its Fc region. So it's really going to be down here, not up here. The fluorescence activated cell sorter detects that fluorescence of your monoclonal antibody. And if the cell fluoresces, it's going to be extracted into a collection tube using an ion beam and electromagnetic separators. So it basically looks like this. There's a big machine with a plastic vial coming out of it, and at the bottom of that is a dripper. And the machine shakes really small vibrations very quickly. That causes a bunch of droplets containing cells to come out one at a time. So these cells They come out one at a time from the dropper, and the computer has a laser that it shines on the cell, and that's either going to lead to fluorescence or it's not going to lead to fluorescence. If the cell fluoresces, that means it has CD4 on its surface, so it's put into a collection vial with other cells expressing CD4. Okay? On the other hand, if the cell doesn't fluoresce, it doesn't have CD4 on its surface, it's going to be put into the waste. This allows you to isolate your CD4 positive cells and throw away all your cells that don't have CD4. Typically, you're then going to take your positive sample that had the CD4 molecules and you're going to rerun it through the fax again just to be sure that you only have CD4 positive cells there, because this machine is not 100% efficient. The machine is capable of processing as many as 100,000 cells per minute. So, very quick and very efficient. You're only going to spend an hour or so with a decent sized sample. I think that's all I want to say here. So you have a cell suspension in a solution. You load it onto the cytometer, the flow cytometer. The individual cells are fluoresced and analyzed by the laser. The machine is set up so that each individual cell enters a single droplet as it leaves the tip of the nozzle. And this is done by vibrating the cytometer. Each drop is given an electronic charge by a laser beam that runs through the droplets right as they come off. And that electronic charge depends on the fluorescence of the cell inside the drop, and it depends on the program settings. So you might want your negative cells to go into the waste, and then your positive cells to go into the collection tube, for example. That would be pretty common. And then once they're given a charge, deflection plates attract or repel the cells accordingly into collection tubes. So you have basically strong electromagnets on either side of the dropping zone, and those electromagnets are going to attract positively charged ones into the collection tube, and they're going to put non-charged ones into the waste for you. Different monoclonal antibodies conjugated to different dyes are going to fluoresce at different wavelengths and therefore can be given different charges and sorted into different tubes. So you can do multiple antibodies when you do this. You can look for multiple surface antigens. I really don't think there's a limit to that. You can probably do like 10 or more surface antigens, and that's not all that uncommon either. And you can do it at the same time. So you can, for example, have CD4 positive, CD8 negative cells in a collection tube. You can have CD8 positive, CD4 negative cells in another tube. And then you can have double positive cells in a third tube. And then the double negative cells can go into your waste. So you can separate cells that are both expressing CD4 and CD8 from those expressing just CD4 and those just expressing CD8. So really cool stuff. The other thing that this can allow you to do is, so it's got these beam splitters that allow it to read the fluorescence at different wavelengths, for example, at a red and green fluorescent wavelength. And then it also looks at the 90-degree light scatter that is not in the plane of the beam. And that allows you to indicate the granularity of the cell. And then forward light scatter is going to allow you to indicate the size of the cell. And so this really allows you to look at cells based on a whole bunch of different dimensions that might be relevant to your experiment. But the point overall is to just separate cells based on phenotype into different vials. Okay. And then the cell characterization function of FACS is called flow cytometry, and a flow cytometry output looks something like this. Here are your CD8 positive cells that don't express CD4. These are your cytotoxic T cells. Here are your CD4 positive cells that don't express CD8. These are your T helper cells. And then here are your double negative cells that are probably not T cells at all, not T helper cells at all, or T cells at all, right? And these might be what you throw away. So you're isolating cytotoxic T cells from T helper cells from non-T cells. I have a video here that you can click on if this doesn't make a whole lot of sense to you. but this is called flow cytometry. So this is just the cell characterization function of the fluorescence activated cell sorter. This part doesn't actually separate them into tubes, although you can probably program it to. This is just a visual representation of your cell populations. And so I'll leave off there.